once more. Thank you. And then we will move on to the next talks. The next two talks are selected from the abstracts. And our next speaker is Hana Valenta from KU Leuven, Belgium. And she will tell us about separation of spectrally overlapping fluorophores using intra-exposure excitation modulation. Excited to hear you.
Thank you. Thank you. Well, first of all, I would like to say that I'm really happy to be here at Elmian, and thanks organizers for giving me the opportunity to give this talk. And I know that you are all tired after yesterday, and I just tried to have your attention for a few more minutes. So first question we can ask is, why would someone use spectrally overlapping fluorophores? Why would someone use the fluorophores of the same color? And the answer is multiplex imaging. Because if we can measure multiple components in the complex system, such as this organoid, but it can be even like single cell can be complex enough, it will help us to understand the complexity of the system, understand how this system works. But also, anyway, in this image, we still have only 3 colors. And why? Because fluorescence microscopy, it's a great tool, but you're still limited by the available spectral window. So the option is to use other properties than the emission color. And I will show you a method we developed in our lab, but I mean, there are also other people who try to use other properties. For example, we can use anisotropy, as it was shown in the lab of Stefan Hell. We can use fluorescence lifetime, as it was shown in the lab of Marcus Sauer, or we can distinguish more probes using light-induced fluorescent dynamics. And there were methods like optical lock-in detection, out-of-phase imaging after optical modulation, or SAPPHIRE. But all these methods with the nice abbreviations, they have a limitation, which is that the fluorescence dynamics has to lie in the temporal resolution of your system. It has to be directly resolved by the camera. And in terms of wide field microscopy, the camera, the exposure and the readout time of your camera will be your limitation. And we were thinking, well, it would be nice to have a method to be able to distinguish also fluorophores who have distinct light-induced dynamics, but they are too quick to be resolved directly. So we tried to develop this method, which I will show you right now. And actually, this method it can enable considerably faster imaging, and it can expand the range of probes we could use for imaging. So one way to use quick dynamics is to use photochromic probes, which includes reversibly switchable fluorescent proteins, which can be turned on and off by different wavelengths. So we can turn them on with the violet wavelength, and we can make them fluorescent, and we can turn them off with the cyan light, with 488, which looks, for example, like this. These are eukaryotic cells expressing reversibly switchable fluorescent protein. So in our method, we want to distinguish distinct light-induced dynamics of spectrally overlapping fluorophores. So I will show you this for green fluorescent proteins. And actually, we will do intra-exposure excitation modulation. And how does it work? So we picked 4 fluorescent proteins that are green: EGFP, FF-DROMPA, FF-DROMPA, FF-DROMPA2F, they are all green. And if you follow me now, so the EGFP is the green one, and when we expose it to the violet light, we will turn it on, and then with the cyan light, we will turn them off. And the EGFP is stable, it's not decreasing its fluorescence intensity, it's not blinking. The FF-DROMPA, which is the dark blue, it's getting turned off. FF-DROMPA FF is the cyan blue, and FF-DROMPA2F is our fastest switcher. And you can see that they have different kinetics, they have different dynamics, they have different, you can turn them on and off with different kinetics. And in our method, we designed illumination pattern when we will acquire only 4 images, which will give us a tool to distinguish these fluorescent proteins. So in the I will walk you through the method now. So for the 4 fluorescent proteins I presented to you, in the image 1, we will have a short pulse of the violet light, and then the cyan light will be on actually all the time. And these lines, with corresponding colors to our fluorescent proteins, this is the theoretical emitted fluorescence I would get. But this is too fast for my camera. So actually for each pattern, the camera will measure just the integrated fluorescence. And this, like this, I will do this for every image. So in the second image, I will just turn them off because I have no violet light. In the image number 3, I will have 3 short pulses to reactivate the fluorescent proteins a bit. And in the last image, I will turn them on completely. So, and for every image, I will get levels of integrated fluorescence, which in the end will lead to the distinct profiles for every fluorescent proteins. And if you look on this graph, it's beautiful because you can see how different they are. And for each image, we have distinct level of fluorescent intensity, which allows us to separate them. So every time we use the image number for normalization, because there they are all in maxima. And for example, if you look at the image number 3, this is perfect to separate the FF-Drompa-F, FF-Drompa-2F, and image number 2, we can separate these two from FF-Drompa and EGFP. And how does it look in practice? So we validated our proof of concept in bacteria, and these are bacteria who express single fluorescent proteins, and you can see that they really blink differently. The fluorescent dynamics is different. So EGFP, there is not much happening, and it's still 4 images. It's only 4 images you need. FF-Drompa, there is a little bit of blinking. FF-Drompa, okay, it stopped. FF-Drompa was blinking more, and yeah, and FF-Drompa-2F is the fastest switcher. So then we were wondering, well, we will just make a mixture and we will separate all these proteins. So we had the fluorescent proteins expressed separately in separate dishes. We made the culture, and then we made the mixture of all these fluorescent proteins, which we embedded in the agar and put on cover slip and do the imaging. And how does it look like? So this was the mixture of the bacteria. So each bacteria expressed different fluorescent proteins. And we applied our method to separate these proteins and classify them to be able to say in which bacteria is which protein. And in the end, we were able to classify quite accurately. So in this map with false colors, you can directly see which bacteria express which protein. And it was using the 4 images, and as you can see in this confusion matrix, that we classify quite accurately the proteins in the bacteria. So this was using the 4 images, but also even if we use only 2 images, it showed potential of our method that you can actually, it can be used with very short exposure times, only using 2 images, which was the image number 1 and image number 4 used for the normalization. The classification was still very accurate. We were always over 95% to say which protein is in which bacteria. This was published. But then you can tell me, well, you showed an organoid and you talk about a cell, but this was still bacteria. But so what about the more complex systems? And my answer is, yes, we can do it, but we need to do a little bit differently. So we will do unmixing per pixel. Because bacteria, it was separation and classification. So this time we will do it per pixel. So we will use the same measurement principle, same reversibly switchable fluorescent proteins, but we will use them in eukaryotic cells, and they will be targeted to different structures. So these are HeLa cells, and these express EGFP targeted to the membrane, FF-Drompa targeted to the nucleus. Then we had FF-Drompa-F targeted to the vimentin, to the cytoskeleton, and in the end, FF-Drompa-2F targeted to the peroxisomes, which are the little dots. And I just wanted to check if the in-cell profiles are the same, the blinking profile is the same under our elimination pattern as in bacteria, because as the cells are complex, there can be something which will influence the behavior of the protein. And it seems that they are following the same pattern. The blinking is the same. because the eGFP, there is nothing much happening, and for example, our fastest protein, which is FF-2F, it follows the same pattern as I showed you before. So this is fine. But I showed you the single fluorophore data, but the fun starts when you have the mixed data. And as you can see in this video, every image, it's still the 4 images, and every image you can see different protein is highlighted, different protein is visible. In the 4th image, it's very clear that there are the dots. Oh, sorry. The dots are. Sorry. The dots are coming out. And in every image, you can see different protein. So we analyze it per pixel, and what I will show you is preliminary data, which we do the analysis with the collaborating group. And we are able, for now, we can show the membrane, which is the eGFP. We can see the fibers of the FF-2F. We can see the peroxisome and the nuclei. But this still needs a little bit of optimization, but we will get there. And so this is to be continued. So what's the take-home message? We developed a fast, very robust method to separate and to, in the end, unmix fluorophores of the same color, fluorophores with the spectral overlap. It's easy to implement. This was all of our wide field microscopy, and it's easy to implement to any microscope you have in the lab, so you don't need very special equipment. And we expand the range of the probes we can use for fluorescence imaging. This is an application which can be a bit deeper, meaning that we can use it not only to fluorescent proteins, but to also organic dyes or even to biosensors. And I just would like to do a little reference to the research we do in the Turku lab that we developed recently in these 2 publications of Tess and Franziska and Anais, biosensors which are photochromic, that they can be switched on and off. And in the end, when I showed this cell, which is cell is very complicated. When I use labeling for example, nucleus, mitochondria, or endoplasmic reticulum, and if these probes will be photochromic, I can use the same color, and I still have, I don't know, red and blue for another structures, and I can do multiplex imaging. I can follow multiple components in a cell. I would like to acknowledge also the authors of this paper, especially Sievert, who left our lab but who worked with me on this paper. And I would like to say thank you to our collaborators from Lille who helped us with the unmixing per pixel. And in the end, as I said, I'm from the Dedeker lab of Professor Peter Dedeker. We are in Leuven. You can follow us on Twitter. And I know the poster session is over, but if you want to know more about our research, you can find posters in the abstract book of number 7, number 21. And I thank you for your attention, and I'm happy to take questions.
Thank you, Hana. That was very interesting. I think we could have time for one question. Over there in the middle. The microphone is coming.
Thank you very much. Fantastic idea.
Thank you.
But it looks like it's just a beginning. Like your 4 patterns of illumination look also relatively crude. Probably if you could do it more comprehensive, and maybe on the detection part, on the detection side, using some kind of artificial intelligence to distinguish between the patterns. What do you think? How many of different fluorophores could you distinguish?
Yeah, that's a good point. We could. Can I still use my slides or not?
You can try. I'm not sure.
Yeah, that's a good point. We could implement artificial intelligence. That's definitely an option. Why not? And to the question, if we can use how many fluorophores we can use, well, we still need to separate them well, right? But if you look at the pattern, for example, we are still able to distinguish FF-2F and FF-2F, which are very, very close. So, I mean, we can go over 4. We can go to 5, 6, even more. It just has to be, we have to be able to separate. But as you can see, even these fast switchers, we are able to separate them.
Thank you. So my gut feeling says that all of them actually any single one, any single two you can distinguish with this approach?
We would love to, yes.
Thanks.
Thank you very much. Let's thank Hana once more.